Hi, my name is Shane,
I'm Dheeraj.
and today we're going to be talking to you about herpes.
So herpes is a virus, and the genome of herpes is DNA. So it's kind of like the same as our cells, where we have a double-stranded piece of DNA in our cells, which give the instructions for different proteins that the virus wants to make, which can help it enter our cells and replicate itself. So it kind of uses us to make more copies of itself, and that's kind of just its main function. Like that's what it's designed to do, is just to make more copies of itself. So this is kind of a picture of what the herpes, like a single herpes virus looks like, called a virion. And as you can see, the blue stuff in the middle is the DNA, and you have all the stuff sticking out of it are proteins that it uses to enter the cell. It has what's called a lipid envelope, which means that it's like a membrane, which is similar to the outsides of our cells. It's consisted of a fatty substance. So if we're talking about the life cycle, the herpes virus enters our cells through receptor-mediated endocytosis, and what that means is that it enters our cell by using one of the proteins that we saw earlier to fit into a receptor on the surface of our cells. So all of our cells are studded with different proteins, and they all look like they all have different shapes and structures. So the one on the virus that matches the one in our cells will bind together, and this will allow our cell to open almost like a door for the virus to enter. And then it uses microtubules to travel to the nucleus. And so the way to think about microtubules is it's kind of like a large ramp or like an escalator in a way, and it's just made out of different different connective tissue and proteins, and that's what helps the virus to travel to the nucleus, also known as the control center of the cell, where replication happens and where the virus can replicate itself using different proteins in its packaging. And so what it'll do is it'll go in and it'll turn its DNA into circular capsids, and it can use this to replicate itself. Also, the virus also blocks production of host cell proteins. It recruits transcription factors from the cell, and it'll degrade host RNA. So what that means is that it'll basically just completely stop our normal cellular processing and stop the proteins we make, and it'll degrade all the DNA and RNA that we're making to make proteins. It'll also use our own machinery to make more copies of itself. So it depends on our machinery to make more copies of itself. Right, so there's 3 different proteins that herpes uses: early proteins, immediate early proteins, and late proteins. And these all help herpes in the different forms of its life, in the different steps of its life cycle, reach the goal of replicating itself. And then before it leaves, it also takes one of the membranes from our cells, and that's where it gets its outer membrane from.
Cool. So there are ways that our body has to help block or get rid of the virus once it's entered. But before it can get into our body, skin is seen kind of like a brick wall on a house, right? So you can't walk into a house unless you're going through an opening door, as Dheeraj talked about earlier, with kind of like the key and the lock mechanism with the different proteins to enter the cell. So our body is able to secrete something called a defensin. And basically what that means is it can insert itself in that fatty acid layer, or that fatty layer that we talked about earlier, and basically kind of create a hole and let the insides leak out. There are cells called dendritic and macrophages that are located and kind of roam around in our skin and underneath our skin. And basically what they're there for is they're there to kind of like a customs checkpoint to see if the cells that are entering the skin or are roaming around the skin have the proper passport. If they don't have that proper passport, they will then eat those cells and then basically bring them to other cells so that they can say, hey, there's a bad guy in our system, and we really need to get rid of this. So what this is here is just what Dheeraj talked about earlier. So as you can see, the little studs on the edge of the fat membrane will kind of connect. The cell will go, oh, those are one of ours, and it will unlock the door and allow the virus to go inside of the cell.
So now I want to talk a little bit about the innate system. So that's the branch of our immune system that isn't specific or has memory capabilities. In other words, it's part of the system that has evolved with us and is more like general. Like it's like the general system that helps stop infections and other kinds of microbes and pathogens that enter our body. So there are these types of receptors called TLR receptors. And what TLR receptors do is that they're on the endosome, the part of so when the virus enters, it enters and goes in through this little like membranous circle called an endosome, and they're like actual receptors. They're like little studded proteins, and those will help detect the virus and detect foreign DNA, and that will send a signal to the cell to start producing, using genes to start producing proteins that will inhibit the virus and the virus's life cycle. And so some of these interferon pathways lead to interferon-stimulated gene products, which are basically just like proteins and other products that will help block infection. It'll stop the virus from making more copies of itself. It'll activate the other part of our immune system, which includes B cells and T cells, the cells that are specific to specific pathogens, like different viruses, like the herpes virus or different types of the herpes virus, and so on and so forth. And it also destroys the RNA the virus makes, and it stops the virus from making proteins. And finally, it also allows the cell to kill itself in case the virus overcomes it. There are also sensors that are outside of the endosome, which we talked about in receptor-mediated endocytosis, and these are just sitting in the cell, like in the free space of the cell called the cytosol. And these receptors also recognize double-stranded DNA, so if the herpes enters a different way, it can also recognize it. And so there are also these cells called dendritic cells that we talked about earlier that eat the virus, and so they'll travel to the lymph nodes, which is sort of like a police station, and they present like parts of the virus that they've cut up. It's kind of like almost like using like fingerprints or an ID tag to show these cells called to show like other cells, and then these other cells that it shows are called T cells, and these are part of the specific immune system, which I talked about, called the adaptive immune system. And these cells are very, very specific. They're almost like assassins in a way. Like they target one type of target, and they can target that one type really well. And so these dendritic cells, or like police officers, show these CD8 T cells which ones they have to go after, and then the T cells go and they take out the virus. And this is called bridging the gap between the innate and adaptive immune system, the two arms of our immune system. And there's also another thing called complement, and what complement is, it's a series of different proteins that are floating throughout the blood, and these if they come in contact with a pathogen like a bacteria or a virus, they can activate a bunch of different effects that can stop the virus or the bacteria, and they can either be activated by themselves or once again activated by receptors. But they aren't very useful for this virus because the virus actually uses one of its proteins to block this pathway.
Cool. So now we're going to start talking about the adaptive immune system. And as D-Raj put, the adaptive immune system, after the immune system has realized that there's an intruder, will notice that key card or that identification and start looking for that specific identification to get it out of our bodies. And so, as we talked about earlier, viruses will need to hijack the systems or the communication systems of our cells. And because of that, they're going to start producing key cards on the surface of our cells. Well, when CD8 positive T cells recognize those key cards are not supposed to be in our body. That identification is not supposed to be in our body. So it will try to then eat and destroy the cell that it finds. So MHC2 is just another type of key card or identification presenting molecule. But the role of this one is, instead of killing, it's kind of like a tattletale. So its job is to tell many different cells across the body, hey, there's an intruder, I need some help, please come help me out. And after being activated, the B cells that we talked about earlier, they'll start to change from cells that are floating around to effector cells. And these effector cells are called plasma cells. Now, effector cells are cells that have been activated, and they're specifically looking for like that identification. And what they'll do is they'll secrete very specific Y-shaped antibodies. And the purpose of this is basically they're wanting to block up the keys that we talked about earlier that are used to kind of unlock the locks of our cells. So what that means is that it will prevent the cell from presenting the key to the locks of our cells, and they will no longer be able to enter. Basically, it's as if we change the locks. And these different proteins, which are not really important to talk about, will help activate what are called natural killer cells. And as their name implies, they're kind of like what D-Raj talked about earlier, is they're just natural assassins. So their job is they will notice things that are not supposed to be in the body, and they'll just start shooting kind of like what you can think is acid at the cell, and just try to completely destroy it in any way that it can.
So now I'm going to talk about what makes herpes so difficult to get rid of, and what makes it stay with you for life. So what HSV does is it establishes latency, which means that it travels from the sites of infection, which are for HSV1, your upper lip. It travels from that area to your peripheral neuron system. So the nerves, the nerves that help you like feel sensations in your lip and under your nose and on your cheeks, it'll go into those neurons, those nerves, and it'll kind of like take a circular piece of its DNA and just kind of sit there next to the nucleus. And so our nervous system, our nerves and our neurons and our brain are known as immunologically privileged sites, which means there isn't an immune response there, because it would be more dangerous to have an immune response there than to just not have one at all, because the immune response causes a lot of inflammation, and this could be damaging to important parts of the brain or the nervous system, which would just create more damage than it would helping if we were infected by something. So as a result, HSV has figured out this really neat trick in order to escape our immune system and stay with us lifelong, because our immune system can't reach these neurons, and whenever we're stressed or if our immune system is suppressed for any reason, or we have other infections, the HSV can reactivate and go back down to the epithelial cells, or the cells in the lip and the mouth, from the neurons that it was hiding in. And so basically, this is what gives HSV, what makes it so difficult to treat, is that we haven't figured out a way to completely get rid of its latency or its ability to hide from the immune system.
Cool. And so basically, this is just kind of a picture of what D-Raj was just saying. And as you can tell, that neuron kind of looks like a highway. And that's basically what it is to HSV. It's a highway that it can travel up and down. And when it starts to notice that there's an immune response and its friends are getting killed, it says, oh, I need to go back, and it will hide in an immunoprivileged site. So our immune system has a very special feature that's called memory. And basically what that means is that once an identification has been presented that's not supposed to be in our cell, it's kind of like they put it on a wanted list. So what it will do is anytime that cell is presented from then on, our immune system will immediately recognize it and say, this needs to be put out of our cells, this needs to be put out of our body. It is not supposed to be here. And basically what this is saying is just, it's just explaining that in a little more complicated way. So the picture that we're trying to paint here with this virus is that the immune system does a very, very, very good job of fighting off this virus. However, because it has that neat trick that D-Raj talked about earlier, it's able to kind of stick with us forever because we don't want to have any kind of friendly fire in your neurons because that would basically affect your brain function. So generally, herpes, the diagnosis, the prognosis is pretty good. It really only is an aesthetic issue for the most part. So the like I said before, the diagnosis, the prognosis looks pretty great. It's something that you can live with. It's something that you can deal with, and it definitely would not affect your everyday life. My name is Shane,
I'm Dheeraj.
and this has been the presentation of herpes.